In Chapter 8, we are going to dive further into the mechanisms and processes of photosynthesis. We've talked previously in Chapter 7 about metabolic processes, and now you all know that metabolic processes in all organisms, from bacteria to humans, require some type of energy. To get this energy, many organisms access stored energy by eating or ingesting other organisms. But where does the stored energy in food originate? All of this energy can actually be traced back to photosynthesis, and we're going to dive further into the processes of photosynthesis in this talk. But before we get into talking about photosynthesis, we have to understand some of the organisms that utilize photosynthesis. And let's begin by talking about the autotrophs and the heterotrophs. So plants, algae, and a group of bacteria known as cyanobacteria, which we can see in figure C right here, are the only organisms capable of performing photosynthesis. Because they use light to manufacture their own food, they are called photoautotrophs. That literally means self-feeders that use light. There are other organisms such as animals and fungi and most bacteria that are known as heterotrophs. Heterotrophs means other feeders, because they have to rely on the sugars produced by photosynthetic organisms for their energy needs. And another third very interesting group of bacteria actually synthesize sugars, and they don't use the sunlight's energy directly, but by extracting energy from inorganic chemical compounds, they do use the processes from the sun's light. So for this reason, they are known as chemoautotrophs or chemiautotrophs. And the chemiautotrophs are the thermophilic bacteria that we see in figure E right here. And they, again, capture energy from inorganic compounds to make food. So what do we see? What do we see in this picture here? We see in figure A, this is just an example of some plants. Figure B, this is the green algae. Figure C is the cyanobacteria. D is actually depicting a deep sea vent, and this is where we find these chemoautotrophs, such as the thermophilic bacteria that we see in figure E. The ecosystem surrounding the vents has a diverse array of animals, such as tube worms, crustaceans, and even octopuses that derive energy from the bacteria that are there. So the bacteria play a very important role in the homeostasis of the ecosystem. And heterotrophs, like the deer that we see in this picture here, and fungi and other bacteria, rely on the sugars produced by the autotrophs for their energy needs. So the energy stored in carbohydrate molecules from photosynthesis passes through the food chain. The predator that eats these deer receives a portion of the energy that originated in the photosynthetic vegetation that the deer once consumed. So here's an overview of photosynthesis, and as you can see in the picture, this is a multi-step process that requires specific wavelengths of visible sunlight. We're going to talk about the wavelengths in the upcoming slides. It also requires carbon dioxide and water as the substrates. And after the process of photosynthesis is complete, what happens is that it releases oxygen and it produces G3P, also known as glyceraldehyde 3-phosphate. Another product is the simple carbohydrate molecules that are very high in energy that can be then converted into glucose, sucrose, or other types of sugar molecules. These sugar molecules contain energy and the energized carbon that all living things need to survive. So in the discussion board this week, I asked you to outline the process of photosynthesis, tell me the reactants, and tell me the products. One thing really important to pay attention to is the waste products in photosynthesis. This is going to be a very important key feature for your discussion board assignment. Oxygen is a waste product, and this could give you a hint as to why plants are able to survive for thousands of years. So just think about that component right there. So before we get into the photosynthetic reaction, we need to understand where all the molecules and compounds actually come from. So everything starts with sunlight and water. Water is absorbed by the roots from the soil. You saw that in the previous picture on the previous slide. Carbon dioxide is acquired from the air. That is what we expel as humans as our byproduct. We breathe out carbon dioxide. And plants obtain it through their stomata. Stoma is the singular form, stomata is the plural form. And these are small pores on the leaf underside. And you can see different stoma depicted in the picture right here. And then again, very important, the waste product of photosynthesis is oxygen, and oxygen leaves through the stomata. This is a really cool video on leaf structure, so please take a minute to pause the PowerPoint, click on this YouTube link, and watch this video. So we've already talked about the photosynthetic reaction, and we have went in depth in the previous chapter about cellular respiration. But I want to re-highlight photosynthesis because repetition is key for memorization and understanding the material. So I'm not going to go back into the stoichiometric balance of this equation because we know this equation is stoichiometrically balanced, but please pay attention to the components, which are the reactants and the products in photosynthesis. So again, in our reactant side, we have carbon dioxide and water. The catalyst is sunlight. That could be a great test question. What's the catalyst for the photosynthetic reaction? And then our products are sugar in the form of glucose, C6H12O6, and oxygen. So just like we talked about the complex metabolic pathways, glycolysis and the TCA cycle and oxidative phosphorylation in cellular respiration, there are complex metabolic pathways in photosynthesis as well. And we're going to highlight these two metabolic pathways. The first one we're going to talk about is the light reaction, and the second one we are going to talk about is known as the Calvin cycle. Photosynthesis takes place in two sequential stages. We have the light-dependent reactions and the light-independent reactions, and it's very important for you all to be able to differentiate between these two types of reactions. So let's start by talking about the light-dependent reaction. What's going to happen here is energy from sunlight is absorbed by chlorophyll, and that energy is converted and stored in the form of chemical energy. And this whole process occurs in the thylakoid membranes of the chloroplast. Very important for you to know where this light reaction occurs. So again, we're converting the light energy to chemical energy, and the products are adenosine triphosphate and NADPH. Really important to know here. This is going to be a test question for sure. It occurs in the thylakoid membranes of the chloroplast. So again, right here in the picture, the thylakoid membranes. We've already talked about the thylakoids. In the light-independent reactions, what's going to happen is the chemical energy harvested during the light-dependent reaction is going to drive the assembly of sugar molecules from carbon dioxide. So although the light-independent reactions do not use light as a reactant, they require the products of the light-dependent reactions to properly function. And there are going to be several enzymes of the light-independent reactions that are going to be activated by light. So light still plays an important role even in the light-independent reactions. The light-dependent reactions utilize certain molecules to temporarily store the energy, and these are referred to as the energy carriers. The energy carriers that move energy from light-dependent reactions to light-independent reactions can be thought as full because they are rich in energy. And after the energy is released, the empty energy carriers are going to return to the light-dependent reaction in order to obtain more energy. And what this picture here is showing is it's highlighting the components inside the chloroplast where the light-dependent and the light-independent reactions are going to take place. Okay, so again, the light-dependent, we're going to be converting light energy to chemical energy. The catalyst is going to be the sun, and then the light-independent, we're going to talk about the Calvin cycle. So the Calvin cycle is going to use the products of the light-dependent reaction. So the Calvin cycle is going to use ATP and the NADPH that was produced by the light-dependent reactions in order to make food or sugar molecules. And again, this reaction occurs in the stroma of the chloroplast. So please don't confuse stroma with stoma and stomata. They are two different components. Please be sure that you understand the definitions of stroma and stoma, as they are two separate entities. Now let's talk in a little more depth about the structure of the chloroplast, where these light-dependent and these light-independent reactions are actually taking place. So the chloroplast is a double membrane structure. There's an outer membrane and there is an inner membrane. So it's a double membrane structure, outer and inner membranes. Consists of stroma. Stroma is the aqueous fluid. Again, don't confuse stroma with stoma and stomata. They are two different things. The stroma is the aqueous layer that is found in the chloroplast. Then we have the grana. These are the stacks of the thylakoids. The granum is plural for grana, so again, this is going to be consisting of stacks of thylakoids, these individual little discs. And the lumen is going to be found inside of the thylakoid. So this picture here is showing the thylakoid stack opened up, and the component inside of the thylakoid is known as the thylakoid lumen. Now the question arises, what actually is light energy? Well, it's electromagnetic energy that's composed of photon particles that travel as waves. So the longer wavelengths, they have further apart crests. So the crest is going to be the high point of the energy wave, and the wavelength is the distance between the crest of the wave. So the wave from crest number 1 to crest number 2. This is going to be what is known as the wavelength. And the bottom of the energy wave is going to be known as the trough. And I'm sure you've all heard the mnemonic before, Roy G. Biv, red, orange, yellow, green, blue, indigo, violet, Roy G. Biv. This is a mnemonic to help you remember the color spectrum. And we're going to talk about the different wavelengths of colors in a little bit. Well, light energy as a whole, we can only see a fraction of this energy because there's only a little bit that's actually in the visible range. A little question for you all here. If you look down at your arms and you see the veins in your arms, what color are your veins? Do they appear blue? Do they appear slightly green? Well, why is that? Do we have blue blood in our body? If we cut ourselves, are we going to bleed blue? This is a question we're going to answer on a future discussion board, so I want you to think about that. Why do our veins in our arms appear blue? I'll give you a little hint. It has something to do with the light energy. Now let's talk about how absorption of light actually happens. So this happens when light energy excites electrons. When a photon of light energy interacts with an electron, the electron may absorb the energy and jump from its lowest energy ground state to an excited state. And that's exactly what you see in the picture here. You see the electron here interacting with the photon, and it goes from the ground state to its excited state. The electron goes to this high energy state because it is photoexcited by the photon. So again, using the mnemonic that I talked about earlier, Roy G. Biv, red, orange, yellow, green, blue, indigo, violet, this will help you understand the light spectrum. And it's very important to understand that the colors of visible light do not carry the same amount of energy. So this V out here that is denoting violet, violet has the shortest wavelength. You can see the distance between the crest of the waves in violet are very, very close together, has the shortest wavelength, compared to a wavelength that is much longer that we see on the opposite end of the color spectrum, like between red and orange. So as violet has the shortest wavelength, it carries the most energy. So again, more energy on this end and less energy on this end. So I can ask you a test question on any one of these colors, and I can ask you which one has more energy and which one has less energy. So it's very important for you to understand the differences in the wavelengths. And wavelengths are measured in a unit known as nanometers. In the visible range from 700 to 400 nanometers, again, violet is going to have the shortest wavelength and the most energy. And red is going to have the longest wavelength and what should it have? The least amount of energy. So again, this color spectrum is really, really important. So X-rays and ultraviolet light, those are going to have the highest types of energy, the highest forms of energy, and infrared light has some of the lowest energy that is associated with the color spectrum. There are different kinds of pigments that exist, and each absorbs only specific wavelengths or colors of visible light. And pigments reflect or transmit the wavelengths that they cannot absorb, which makes them appear as a mixture of the reflected or transmitted colors. So please understand that. That's going to give you the answer to the question I asked on the previous slide about why our veins appear blue to us. It's not that we have blue blood. It has something to do with the color spectrum, and I just gave you the answer in that previous statement. So if you didn't catch it, pause the video, rewind to the beginning of this slide, and write down in detail what I just said, and come up with a logical explanation of why our veins appear blue. So now let's dive further into the main pigments of the thylakoid membranes. So we have chlorophyll A, chlorophyll B, chlorophyll C, and there's also another type known as D that's found in prokaryotes that we call bacterial chlorophyll. But we're going to focus on A, B, and C in this slide, and C can also be termed beta carotene. Okay, so chlorophyll A and chlorophyll B are really important for capturing light for photosynthesis, and this is the reason leaves are green, because the green wavelengths are reflected. That is what we see. We see the wavelengths that are reflected. Beta carotene helps to protect photosystems by dissipating excess energy. And beta carotene is found in carrots and oranges and helps to give them that orange appearance. And there's other carotenoids such as lycopene that gives the tomatoes that we eat their red appearance. Or zeaxanthin that gives the yellow appearance to corn and corn seeds. This is a really cool video on the light reaction. So again, please take some time to watch this, pause the lecture at this point, and click on the YouTube video and watch this video on the light reaction. Now let's talk about how these light-dependent reactions actually work. So the overall function of these reactions is to convert solar energy into chemical energy in the form of NADPH and ATP. And then remember, this chemical energy is going to be used to support the light-independent reactions and fuels the assembly of sugar molecules. So what we see in the picture right here is the series of the light-dependent reactions. So protein complexes and pigment molecules work together to produce NADPH and ATP. So you can see some of the pigment molecules represented by these green circles here. The numbering of the photosystems is a little unique, and it's numbered in order of discovery, not the order of the transfer of electrons. So photosystem 2 was actually discovered before photosystem 1. So that's why the numbering system goes 2 to 1, not 1 to 2. So photosystem 2 and photosystem 1 both consist of a light harvesting complex and a reaction center. Pigments in the light harvesting complex pass light energy to two special chlorophyll A molecules in the reaction center. So what's going to happen in this reaction here is the photon is going to be emitted, and it is going to come in to the photosystem, and it is going to interact with these pigment molecules. The pigment molecules are going to transfer the energy to other pigment molecules, and then eventually it's going to come into contact with these special pairs of chlorophyll A molecules in the reaction center. And then what's going to happen in the reaction center is the light is going to excite electrons from the chlorophyll A pair, and those are going to be passed on to the first electron acceptor of the electron transport chain. So right here, the electrons are going to be excited, and then they're going to pass on the energy to the first electron acceptor, that's depicted right there, of the electron transport chain. This is a light-driven redox reaction. So remember, review your redox reactions. What does that mean? The lost electron is going to be replaced. And down here, we're going to talk about what happens to the electrons in photosystems 2 and photosystem 1. So in photosystem 2, the electron comes from the splitting of water, which releases oxygen as a waste product. Again, that's really important to know with photosynthesis as a whole, that oxygen is a waste product. In photosystem 1, the electron comes from the electron transport chain. So again, this is a really great test question and quiz question. Where do the electrons come from in the different photosystem complexes? In photosystem 2, the electron comes from the splitting of water, and in photosystem 1, the electron comes from the electron transport chain. Really important to know. There are two parts of the electron transport chain in the light reaction. Let's begin by talking about photosystem 2 up here. So in photosystem 2, in the reaction center, energy from sunlight is used to extract electrons from water. The electrons are going to travel through the chloroplast, etc., to the photosystem 1 eventually. So they're going to travel down here through the chloroplast, and eventually they're going to get over here into photosystem 1. And what happens in photosystem 1 is going to be the reduction of NADP to NADPH. So that's via the enzyme NADP reductase. So that's going to turn NADP into NADPH. The electron transport chain moves protons across the thylakoid membrane into the lumen. And at the same time, that splitting of water is going to add protons to the lumen, and the reduction of NADPH removes protons from the stroma. And what is the net result? The net result is going to be a low pH in the thylakoid lumen and a high pH in the stroma. ATP synthase, as depicted down here, uses this electrochemical gradient to make ATP. Just like the electron transport chain that we talked about in Chapter 7 dealing with cellular respiration, there is going to be a hydrogen gradient that's created in the electron transport chain in plants as electrons fall down the chain. And important to note on this slide, ATP synthase, which is depicted down here in the picture, uses this gradient to generate ATP. And this is done through a process known as chemiosmosis. What we see here is ADP is gathering up that free phosphate group and generating ATP. And we're going to talk in depth about ATP and its important role in the facilitation of the Calvin cycle. And also remember what we talked about with the formation of NADPH. So NADPH and ATP are going to be two really important players that help to facilitate the Calvin cycle, which we're going to talk about on the upcoming slides. And again, just to highlight the importance of this, when I reiterate concepts over and over again, that is for a reason, most likely going to be a test question. So again, the ATP and the NADPH that is generated from this light-dependent reaction, where are they made? They're made in the stroma, and they're going to be used for the Calvin cycle. So again, ATP, NADPH, they are generated in the stroma. They are part of the light-dependent reaction. Remember, the light-independent reactions do occur without light, but they're using the energy and the resources from this original light-dependent reaction in order to facilitate and accelerate the Calvin cycle. Now we're going to talk a little more about the Calvin cycle. On this slide, I'm just going to give you some background information, and on the next slide, we're going to get into what is actually happening in the different phases. So in plants, carbon dioxide, as you know, we've talked about this already, it's going to enter the leaves through the stomata. And once it's in the stomata, it's going to diffuse over short distances through intercellular spaces until it reaches the mesophyll cells. And once it reaches the mesophyll cells, then the carbon dioxide is going to diffuse into the stroma of the chloroplast. And remember, the stroma is the location for the light-independent reactions of photosynthesis. So that's what we're going to be talking about here, the light-independent reactions. So these reactions have several names associated with them. So the light-independent reactions are known as the Calvin cycle, which is actually named after the scientist who discovered it. And because this occurs in a cyclic fashion, it is termed the Calvin cycle. Others may call it the Calvin-Benson cycle to include the other scientist that was involved in the discovery of it. And a very outdated name, one that's not really used anymore, and please don't use it for this class, but it's important to know the general background information about it. This is sometimes called the dark cycle because energy is not directly required for this. But as you know, the NADPH and the ATP that was generated from the light-dependent reactions plays an integral role in facilitating the Calvin cycle. So even though there's not a light requirement in these light-independent reactions, there was the initial input of light from the light-dependent reactions that was necessary in order to make everything occur in those light-dependent reactions to start the light-independent reaction cycle. All right, now we're going to dive into the different phases of the Calvin cycle, and it's really important for you all to be able to differentiate these three phases and understand what's going on in them. We're going to talk about phase 1 first, or carbon fixation. So before we talk about this reaction, I want to define a few things. So I want to define the enzyme Rubisco first, and I call this enzyme Rubisco because it has a very long name. The enzyme is actually ribulose 1,5-bisphosphate carboxylase oxygenase. So that's a tongue twister. We're going to call that Rubisco. And I'm also going to abbreviate RUBP, ribulose bisphosphate. So what's happening in phase 1 is carbon dioxide is going to be added to RUBP by the enzyme Rubisco, and 2,3-phosphoglycerate molecules are going to be produced. Next is going to be phase 2, also known as reduction. In phase 2, what's going to happen is that ATP and the NADPH that we've acquired from those light-dependent reactions are going to be used to add electrons and to make sugar, or make glyceraldehyde 3-phosphate. And then finally, in phase 3, what's going to happen is we are going to regenerate the RUBP. And in phase 3, the RUBP molecule that starts the cycle is regenerated so that the cycle can continue. Only one carbon dioxide molecule is incorporated at a time, so the cycle must be completed 3 times to produce a single 3-carbon 3-GP molecule, and 6 times to produce a 6-carbon glucose molecule. And this slide here is a really cool animation of the entire process which we've discussed throughout this lecture. So this is the last slide. Please take some time to pause the lecture and review this video as well.